Hello everyone. Welcome to today's live broadcast, Practical Considerations for Fluorescence and Imaging. I'm Julianne Chide of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Thermo Fisher Scientific is the world leader in serving science. Our mission is to enable our customers to make the world healthier, cleaner, and safer. We help our customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. Through our premier brands, Thermo Scientific Applied Biosystems, Invitrogen, Fisher Scientific, and Unity Lab Services, we offer an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. For more information, please visit www.thermofisher.com. We have a few important announcements before we begin. The webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button on the lower left. I would now like to introduce today's speaker, Trillium Blackmer. Trillium Blackmer is a senior staff scientist in the Biosciences Division of Thermo Fisher Scientific, located in Oregon. For the past 7 years, she has worked in product development for fluorescence-based probes and has also been the lead writer and developer of an educational resource for fluorescence imaging basics, the Molecular Approach, School of Fluorescence. She received her BS in chemistry from the University of San Francisco and her PhD in molecular pharmacology and toxicology from Northwestern University Medical School. She served in postdoctoral positions in neuroscience labs specializing in electrophysiology and fluorescence microscopy before moving over to biotechnology startup in 2008. Her namesake is a woodland flower, not a chemical. I will now turn it over to Trillium Blackmer for a presentation.
Hello everyone and welcome. Today I am going to go over a few things you can do to improve your imaging experiments and also tell you about some different reagents that can help in experimental design and also in choosing what reagents you might want to use to ask for your scientific questions. So the three main things that I'd like to cover today are go over some of the basic reasons you might want to use fluorescence in your microscopy experiments, bring up some common problems that both experts and beginners run into, and different ways that you can solve those problems. Science is mostly about iteration. It's about 1% inspiration, 99% iteration. So we'll go over some tips and tricks, and then reagents and instruments that can also help with some of those problems. And then lastly, if I don't talk too much, I'll mention a few online resources that Thermo Fisher has that can give you some of the material you see today, and also some apps for your phone that can help with experimental design and protocols. So a brief history of molecular probes. It was founded by a chemist in Minnesota in his kitchen. And then they moved to Texas in the late 70s and started a company there. And you'll see some of the place names from where he moved around the country in the names of the dyes, for instance, Texas Red. And then he relocated to Oregon and a place near Eugene, which is where we're located now. And so you'll sometimes see some of the names, like Oregon Green is a common dye name that Molecular Probes catalog contains. But really the more famous dyes and probes that we're known for were developed in the 90s, and those include the mito trackers, lyso trackers, Flow 4AM, things like that, and we'll go over some of them today. We've gone through a few different parent corporation name changes. Invitrogen acquired molecular probes and then merged with ABI, and we were known as Life Technologies. But now we've been acquired by Thermo Fisher, and that's our parent corporation. Those phone numbers you see on your screen are phone numbers. The 800 number is directly to Molecular Probes Technical Support, and the 541 number is if you wanted to talk to a real person at our front desk. So why do we use fluorescence in microscopy? One of the big reasons we use it is for resolution. The modern CCD cameras that acquire the images that we usually acquire in fluorescence microscopy have a much bigger range in their both the wavelength and their sensitivity. So what you see here is under the blue peak is what the human eye can generally see. If you're young, you can see all the way out to 700 nanometers. Usually older eyes like mine can't even see that far, and it cuts off somewhere in the 600s. But you can see that CCD cameras can see a much wider range of wavelengths, and that helps. And they're also much more efficient in terms of how many photons they need to detect a signal. So resolution is one of the main reasons we would use fluorescence microscopy. Another reason is selectivity. So what you can see in this slide is the same fluorophore, which is that green blob labeled fluorophore, and it's conjugated to different things, and that's what provides specificity for where it goes in the cell. So in the first example, this fluorophore is conjugated to a molecule that targets actin, and you can see an example of what it looks like when you label actin in green in the little picture below. In the middle example, the fluorophore has been conjugated to an antibody, and then you can see that usually they're secondary antibodies, and you can see in the picture below what's happened when you've used immunolabeling to deliver that fluorescent signal. And then the last example, which is a little less common today, but it still gets used, is where you deliver the fluorophore to the internal area of the cell, maybe by injection, and that's usually when the fluorophore is in a salt form, and you can look at cell processes and how they grow. So those are all the same fluorophores, and they just change in their selectivity with how they're conjugated or how they're applied to the cell. So that's another really convenient feature for fluorescence microscopy. Another big one, which you're seeing in the slide here, is the contrast that fluorescence labeling provides. So when you look at that transmitted light image, you can see that you get some detail, but not a lot. And if you label the nucleus and the actin filaments with fluorescent labels, you get a lot more contrast between them and their surroundings. So you get a better picture of the structures inside and outside of the cell when you use fluorescent labels. So, for example, this is just another picture that shows you what something looks like. These cells are proliferating, and this is in bright field. And then if you were to look at it with labels delivered to the Golgi and the nucleus, you can see that the nucleus is here in green, the Golgi is in red, and you get a lot more information than you did just with bright field. So with any advantage, there always are costs associated with it, and science is no exception. Some common issues that people run into when they use fluorescent microscopy can include background fluorescence. We're going to spend quite a bit of time talking about ways to improve your signal to background, which dyes and reagents to choose. Sometimes you might run into problems where your cells seem to be dying once you put them on the microscope, or you might have experimental questions about how many cells are dead, how many are alive. We'll go over some reagents that can help there. And then there's also issues of phototoxicity. We're going to explain what phototoxicity is and some ways to avoid it. Same with photo bleaching and other common problems. So signal to background is really the name of the game when it comes to a successful fluorescent imaging experiment. And the way we define signal and background is we say signal is the fluorescent probe giving you a signal from where you want it to be. So if you have a specific probe, it's the fluorescence you want to see. Background is basically fluorescence from everywhere else, and it can be mostly non-specific background fluorescence, and it can come from a variety of places in your experiment. And that's what this little cartoon is showing you. So it can come from the media that your cells are in when you do your imaging experiment. It can come from the vessel. It can come sometimes from the reagents that you use, or even your sample. This is especially problematic in tissue where it has a lot of autofluorescence. So we'll briefly go over a few different ways to kind of work around the background fluorescence in each of these areas so that your signal is nice and bright and you can do a successful experiment. First of all, before you get started, just make sure your microscope is functioning properly. That might be really basic, but it's key. And what you're seeing here, you might think, oh, you're looking at these blue blobs are nuclei that are labeled with a fluorescent probe, Hertz. And you might think that over there in the corner where it's very dim, that's just a really low signal, and somehow they were labeled unevenly, but that's not the case. What happened here was that the microscope was not in alignment, and so this meant that the picture looked dimmer in some areas and brighter in others, and it was due to just the light didn't have a clear path. So make sure your microscope's functioning properly, and if you have a question about it, if you're just beginning, you might ask somebody else. Look at something that you think should have a really good label. Make sure that all the field of view has the same amount of signal before you start to troubleshoot your actual experimental design. Another thing that people don't always like to hear, but it's actually pretty important, is that usually when you purchase a dye to use on your cells, a range of concentrations are given. And that's because the optimal concentration for your particular cell or tissue may not be the same as someone else's. So what you really need to do if you're having problems with signal and background is to optimize your dye concentration. And what you're seeing here in this series of panels is that concentration can really matter. The one that's labeled 1x is the optimal amount of signal to background, and you can see in the next panel, which should be labeled 4x. I apologize, somehow the concentrations got changed on the bottom there. But in the bottom panel there, it looks like your signal is brighter, but the background has also increased. So in fact, it's not the best concentration to be using. And then if you go all the way up to 12x, you can see that the background's even visible to your eye, not just to the camera, and that would not be a good concentration to use either. So optimizing your dye concentration, checking a range of concentrations can be helpful if you're running into either low signal or really high background. Another thing to do when you're in the experimental design portion of your experiment is make sure that your fluorophores are the best match to the filters on your microscope, and that can prevent what we refer to as bleed through. And what you're seeing here in this black and white picture is a label that was used, and it was intended to be to label something that would show up in the TRITC channel. And what happened was, because the dye had such a broad emission spectrum, it actually did what we call bleed through to another channel, the FITC channel, and it's not pseudo-colored, but you're just having a look at it from just the image that the camera records, and you can see that those actin filaments are actually showing up in a channel they weren't intended to, and that could also give you background fluorescence when in an area you didn't want. So in the next slide, Just a reminder that if you are using immunolabeling where you use antibodies, generally in imaging you use a primary antibody against your epitope and then a secondary antibody that has the fluorophore conjugated on it. And you just want to avoid having more than one primary antibody raised in the same host species if you're doing multicolor with many different antibodies. In the next slide, I'm showing you a few different pictures of imaging vessels. And so if you want your best cover of the journal quality fluorescent image, you're going to want to use glass. And that can be challenging sometimes. Cells don't always like to grow on glass. And when you're doing live cells, you want your cells happy as well. So that you have to be the best judge of when you want to use the glass. I mean, a lot of times if you look at that orange lidded flask, many times you're checking cells in the actual tissue culture vessel they're commonly grown in. That plastic is fine for growing cells, it's great, and it will work for quick checks, but if you want a really high quality image, you're going to have to culture your cells on glass. And that's because if you look in the little cartoon, at high magnification and high resolution, the glass is the best match for immersion oils that a lot of high magnification objectives use, because the refractive index is matched, the oil to the glass, to the optics in your objective. So you'll just get the best image there if you use a glass vessel. And some ones I really like to use are these little round ones that you see. So they're plastic, which makes it easier to handle, but then they have a glass bottom in them, and you can see that those milk ones are quite handy. Another thing to use if you're trying to do a lot of experiments at the same time is a 96 well plate that you can see in the middle there in that top panel. And just some things to remember there: if you're using 96 well plates, you want to use probably black plastic because it tends to have less autofluorescence than white, and you want to use glass bottom ones. That'll give you a better image. So kind of changing tact a little bit, if you've taken care of the instrument and the culturing vessel, another thing to think about is what solution your cells are being imaged in if you're doing a live cell experiment. And something to consider is to not image them in their culturing media, because culturing media tends to have a lot of fluorescent compounds in it that will give you background when you're trying to look at your specific fluorescent signal. So one thing that you can use is something that we sell called a live cell imaging solution. It's a product that I developed, and it's based on Ringer's solution. Ringer's is a very old recipe that physiologists have used since the 1800s. It's also what you get if you go to the hospital and you have an IV. It's very related to that drip that goes into your blood. It's very physiological. The nice thing about it is it's optically clear, and it will keep your cells very healthy while they're on the microscope. But the caveat there is that it's really best for experiments that get done in 4 hours or less. If you want something where you're following an experiment, you're following cells out, and you want to image them, for some reason it's important that you image them in the same media that they were cultured in, then the suggestion there is to use a DMEM media that was developed to be as optically inert as possible, and that's that flow bright media that you're seeing on the slide there. What you can see from the data there, it supports proliferation of cells, so they proliferate and grow normally, but the background fluorescence is much lower than DMEM. It's comparable to PBS or another saline solution. And so your signal to noise is comparable to PBS, which you don't get when you image in DMEM or other medias like that, but it can also support proliferation and normal cell function that goes on over a process of 4 hours or longer. And so then in the next slide, what we're kind of addressing now are all the different questions you can ask about how healthy your cells are. And you may be asking these questions because you are studying a process that involves cell viability or proliferation or apoptosis, or you might be asking these questions because you want to make sure that your experimental technique isn't changing or biasing your results in terms of, are your cells still healthy when they're on the microscope and you're studying them? And so something to understand is really, when we talk about fluorescent imaging, what we mean by a live cell and what we mean by a dead cell. So what makes a cell alive is a philosophical question, really, and it could be debated and has been. But when it comes to fluorescent reagents, the way we define a live cell is a cell whose membrane is intact and it has enzymes that can support the cleavage of a dye that, when it's in its native form, it's not fluorescent, but then by enzymes that all cells contain, something gets cleaved off and it becomes fluorescent. So that we take as our live readout. And then for a dead readout, that would be the case from a basic fluorescent imaging perspective, where a impermeant nucleic acid, so it normally cannot cross a healthy cell membrane, but when cells die, their membranes become permeant. So you would get a signal from your nucleus if you were using a dead cell indicator that showed that that nucleic acid was able to bind DNA and fluoresce, and it couldn't do that if the cell membrane was healthy and intact. So that's kind of important to keep in mind when you're thinking about live cell indicators and dead cell indicators. Live cell indicators are things that usually rely on an enzyme substrate in your cell, and dead cell indicators are usually things that are impermeant nucleic acid dyes that are able to get to the nucleic acid in the cell's nucleus because the cell is sick or dying and its membrane is no longer intact. So one thing that people run into problems with is a lot of live dead imaging kits don't fit into filter sets very well there because they're based on flow cytometry, who tend to have a different filter setup. But we do have one that's ideal for FITSI and Texas Red filters, and you can see this beautiful image of a green live cells. That means the enzyme is active in those cells, and so they're fluorescing green, and the dead cells where the membranes are permeant to nucleic acid dyes are showing up as those red nuclei. And I think that in the next movie that's played, you can actually see a movie of how the cells would look if you were looking in the ocular and they started out healthy and then died. So what you saw there was that the cells started out green and healthy. That was their live cell indication. And then what we did at the microscope is we actually poisoned them. We pipetted in a bunch of DMSO that basically blew up their membranes and killed them, and you saw that loss of green fluorescence and then the red indication that the cells were officially dead. And so that's kind of the sort of thing, that was a very dramatic example, but that's the sort of thing that you could expect to see if you used a live dead imaging kit with those FITSI and Texas red filter selections. So in the next slide, we're going to explain a little bit about indicators for apoptosis, which is another particular way that cells can die is through an apoptotic pathway. And CellEvent Caspase 3.7 is a reagent that it's dark, it has no fluorescence until an enzyme is activated in the apoptotic pathway, Caspase 3.7, that's the enzyme. And when that happens, it cleaves off a peptide off of this dye, and then it causes the dye, which is a DNA dye, to be free and able to bind to DNA. And so when you cleave off that peptide, the DNA dye can go and bind to DNA, and what you see in no treatment is dark, but when the cells have been induced to become apoptotic with staurosporin, what you see there is that you get that green indicator that tells you those cells are undergoing apoptosis, and it's a readout in particular of the Caspase 3.7 pathway. So if you wanted to multiplex it with something like TMRM, TMRM is a mitochondrial health readout, and you can see at 0 hour the cells are labeled with red. Those are those red spots you see, and that means their mitochondria are all intact and functioning. But when the cells are treated with staurosporin to evoke apoptosis, you can see that by about 3 hours, the red signal's all gone because the mitochondria are no longer healthy, so they've lost that signal of mitochondrial health. And by 7 hours, most of the cells are green, showing that they've undergone apoptosis, and that's that CellEvent signal. So that CellEvent and a few of our other reagents are actually available in a dropper bottle format that you can keep on your lab bench, and all the protocols are the same. You just drop in 2 drops per mil, and you can look for those under the Ready Probes reagent. If you search for that, you'll find that on our website, and that includes CellEvent and some of the other nucleic acid dyes. So another common question when it comes to cell health is oxidative stress. Oxidative stress is reactive oxygen species that get generated through a variety of ways. And we have a few really nice probes to ask questions about whether your cells are undergoing oxidative stress or not. The one you're looking at here is CellRox Deep Red. And what you can see on the top panel are the nuclei in control. Those are blue nuclei. And then what you see when the cells are treated with something that evokes hypoxia, and so when they're in hypoxia, they undergo that oxidative stress, and they generate a lot of reactive oxygen species. So then you see that little pinkish red signal show up. And what you see in the bottom panel is that that signal is retained if you want to fix your cells afterwards, which makes it convenient to do a live cell imaging experiment and then fix it and retain the signal. And you can also this is a far red sensor, which is nice when you're working in tissue or other things that the green channel is being used. But they're also available in green and orange wavelengths as well. And I think that the next slide is a movie where you can see CellRox green. So what you'll see is that the cells will start out with no color, but as they undergo oxidative stress, they'll become green. So that happened pretty fast, but hopefully you got the idea. It's sometimes nice to see them instead of just pictures, to see the kind of time frame you're talking about in a live image. So another common question to ask about your cells or tissue is what's proliferating. And EDU is a pretty common way. You use a modified uracil that labels DNA while DNA is being made, and that would be while your cells are proliferating. And then it gets incorporated into the DNA, and you can label that special, those special sugars that got incorporated into the DNA while the cells were proliferating. And the nice thing about the product that we call ClickIt EDU is that it's not radioactive, so you can look at proliferation fluorescently, and it's really a nice, simple protocol, because you can see here what ClickIt EDU takes the place of is what's referred to as a BRDU protocol, and that can take all day. Whereas the ClickIt EDU is nice because you feed the cells, they proliferate, and then the actual hands-on work is about an hour, and you get a really nice readout in fluorescent microscopy or flow or high throughput imaging of which cells are proliferating and which are not. But one of the issues with ClickIt was that a lot of people like to use GFP, green fluorescent protein, to label structures in their cell. And with classic BRDU and ClickIt EDU, what you see there in that upper panel is that you lose your GFP signal. It's due to the chemical components of the reactions where you label that DNA. It kills the GFP signal. So a lot of customers talked to us and said, you know, we really would like to be able to look at proliferation using this non-radioactive quick way, but we want to be able to see our GFP signal as well. So a colleague of mine developed ClickIt Plus EDU, and that's where the reaction has been reformulated. It preserves the GFP signal and the EDU signal, and that's what you're seeing in that third panel. You're seeing the nuclei in blue. It's a little hard to see, but I think you can see them. And then you're seeing the GFP labeled cells in green, and you're seeing the cells that were proliferating at the time they were fixed in red. So you can see you get all of the signals you want, whereas in the other two panels, the BRDU and the classic ClickIt, you lost your GFP signal. That last panel is just a control showing you that the ClickIt signal is in fact real, and if you take away the copper that's necessary to catalyze that reaction, then you lose your EDU signal. So a nice thing about ClickIt is that it's a very specific readout of cell proliferation, and it works across almost every species that's been tested. And you're seeing just a nice example of a bunch of different species here, from zebrafish to mouse, and so chicks, E. coli. It basically has worked in almost every species and kingdom that's been tested in. So this slide is here to remind me that to talk about once your cells are out of the incubator and you're putting them on the stage of the microscope to do your live cell imaging experiment, it can be a very harsh environment for cells. They're used to being in the incubator, they're used to their media being a certain pH, they're used to a certain amount of CO2 and oxygen. And so when you take them out of that incubator and put them on the microscope, you have to be sure to do some things to accommodate them and control their environment. And because if you don't, it's kind of like asking you to get on that frozen bicycle and start riding like you were riding on a nice summer day. and we can imagine how that would go. So one of the problems, this slide always kind of makes me a little sad as an electrophysiologist who did 2-photon imaging, but it's true. There are usually a lot of steps involved in doing high-resolution live cell imaging. I think this is 11 or 12 steps, and people can get pretty intimidated using it and trying to keep your cells healthy and do all of the things you need to do to use this instrument. So there are instruments that, to be honest, I was a little skeptical of when I first started using them, but now I love them and I'm a big fan. These are digital microscopes that are really easy to use. There's not 11 steps or 12 steps to turn it on. You can do phase, you can do fluorescence microscopy, and it's nice because they're LED powered, so you have less photo bleaching in your experiments, which is good. And one of my favorite things about them, my favorite one is the FL Auto, and the reason I love it is because I'm a physiologist, and I love that you can get an Onstage incubator that's pretty nice and simple to use, and that gets us back to, that's why we're going here. You can put your cells in that nice controlled environment. So you can regulate the temperature, the humidity, how much oxygen they're getting, and it's all right there on the stage of the microscope, and you can control it digitally while you're setting up your experiment, and it's a really nice feature. I even like it better now than my 12-step confocal. But something else that's worth mentioning just about the FL Auto is that you can get more than one kind of camera. So you can get color and monochrome cameras, and that can be really important when you're imaging tissue. And then it also does what we call image stitching, where it takes a series of images of something really big and can stitch them together to give you a complete image of something at a magnification that normally you couldn't collect all the images of. And that's really taking advantage of the fact that it's a digital microscope. But now back to the hard stuff. So one of the challenges of live cell imaging is phototoxicity, and that is something that you'll see if you do live cell imaging, and you kind of have to learn about it the hard way. What you can see here, this is in a couple different examples. What you can see on the top panel, that gray and green image, is that if you look to the right, where it says illuminated area, you can see that the green signal is pretty dim, and I don't know if you can see it at the resolution on your computer, but you'll have to take my word for it. The cells are really unhealthy looking. The membranes look terrible. Probably if you had a dead cell indicator in there, it would be screaming red because those cells have all died. And what you see in the non-illuminated area is that you have a nice bright green fluorescent signal from your GFP labeled nuclei, and the cells are happy and thriving. And sometimes people think that that's from the expression of a probe or labeling with a dye. It's really not. It's from the actual light that you shine on your cells to get the very images you want can be toxic to cells. And that's because most cells are not intended to ever see light the way we do when we interrogate them on a microscope. So it's something to be aware of. And if you look in the bottom panel, you're looking at some fibroblast cells that had some scratch wounds scratched across them, and they're labeled with cell tracker deep red. And then it's just showing you, and you're going to have to ignore the titles on the bottom there because they got flipped, and I'm sorry. But what you're looking at in that scratch, where there's basically a tunnel through the cells are shown in red. And they didn't ever grow back into that scratch wound, and that's because they were under illumination. So they had undergone time-lapse imaging for 10 hours. And it wasn't the probe itself that caused that phototoxicity, because if you look in the non-illuminated area, the cells have grown back into the wound, and you can see that they're proliferating and they're healthy. So that's phototoxicity, and what that is it's cell death that's induced by light exposure. It tends to be independent of fluorescent labels. They can contribute to some of the toxicity associated with light, but you can cause your cells harm just by shining light on them with no fluorescent label at all. And the things you should watch out for when you're doing your live cell imaging experiment are seeing cells round up and detach, float off of the culturing vessel. If you're used to watching them move and grow, and that movement and growth slows. And where you tend to see these problems is when you're doing live cells, when you're doing time lapses that are over long periods of time. So what can you do to get around that? Well, first of all, you can make sure that your cell environment is as comfortable for the cell as possible, and that goes back to making sure that it's in, a solution that's physiological, it's the right pH, it's warm, you have that Onstage incubator to keep your cells happy. But then when it comes to actually doing the imaging, there's also a few things to keep in mind if you see phototoxicity. You could use shorter exposure times. And so, a lot of times we want to expose for as long as possible to get the highest quality image. But when it comes to time-lapse microscopy, you might need to cut back on your exposure time so that the amount of light that those cells see over time is lessened. You also, as much as autofocus is nice to use to find your focal plane, you probably don't want to do your time-lapse where you've used the autofocus. You want to minimize that as much as possible for those overnight or long time-lapse experiments, because autofocusing uses a lot of light. So the cells only can have so much light shined on them, and if you use that up with your autofocus, by the time you do your actual experiment, the cells will be pretty unhealthy. Another thing to do is to use optimal filters, and we talked about that a little bit. You want to make sure that you're using the right filter set so that you're harvesting as much of the signal as possible. It'll mean you can use shorter exposure times. You also might think about, if you're trying to image overnight or a whole day, might think, and you see phototoxicity, you might want to think about increasing the interval between the times you take images. So not only shortening the amount of time that you expose the cells to light and get your image, but also lengthening the amount of time in between images in your time lapse. And the last thing you can do is around dye selection. So basically, the brighter the dye, the shorter the exposure time, and a dye matched to its filter will also allow you to have much shorter exposure times. So what are some common really bright dyes that are good to use when we're trying to do cell biology live cell imaging? Organelle Tracker dyes are nice because they are very bright and specific. You can see them here labeling the Golgi in red, and in each case the nucleus is labeled in blue. Lysosomes can be labeled using these tracker dyes. Mitochondria, we saw TMRM with the mitochondrial cell health indicator. And then the ER can be labeled as well. So those are nice bright reagents to try. Another thing to think about when you're designing your experiment is that if you've got autofluorescence or you're having problems with phototoxicity, you can try imaging out in longer wavelengths. Those have less energy associated with them, and sometimes they can help with your phototoxicity issues. So here, just for example, is Lyso Tracker, and it's in a deep red, so that would be a longer wavelength red indicator. Another way to label cells organelles and structures is to use genetically encoded labels, and that takes advantage of fluorescent proteins. And you can send the fluorescent proteins to different structures in the cell. The hard part about that is how do you get that DNA into the cell to start with in order to get your structures labeled with the fluorescent proteins. And a really nice way to do it is to use the baculovirus system. And it's not actually a viral infection because it's an insect virus, so it's non-toxic to cells. It's labeled biosafety level 1. So if you're doing cell culture, you can do this. And it's not replicating in mammalian cells. But what it does is it delivers the DNA into the cell, but then the DNA has a mammalian promoter. So the virus, the baculovirus, is not able to replicate, and the expression of the fluorescent protein targeted to these various structures is driven by a mammalian promoter. And it's really nice because you can have a really nice rate of expression in traditionally hard to transfect cells like primary cells, stem cells, neurons. And you basically pipette it into your your plate of cells, and you incubate it overnight, and then by the next day you should have labeled structures. And sometimes it does take a little titration, right? That goes back to optimizing your label for your specific cell type. And so what we're showing here though is that you get a really nice high rate of transduction with different targeted GFP, tubulin, mitochondrial, and early endosome. And then on the right there, we're showing just different traditionally difficult to transfect cell lines that show nice bright signals. You even can see hippocampal neurons. So we talked about reagents a little bit and reagent selection. We talked about different questions you can ask of your cells, and we talked about media to image your cells in and vessels that could help cut down on your background fluorescence. Sometimes you do all of that and you still have a signal that's being very challenging and it's really hard to see above all the background. There are products like this one. It's Backdrop. It's a background suppressor, and it works as a chemical filter to suppress the fluorescence outside a cell. So this reagent is just intended for live cells. It's only intended for internal targets, and you have to kind of be brave when you use it, because when you drop it into your media, it will change the color of your media. It's basically a chemical filter on your media, but you have to keep going and add enough, because if you stop halfway, it actually just makes the background worse. But if you add enough in the channel you want to suppress your background, you will be pleasantly surprised. And you can kind of see that in the bottom panel there with the plus sign, and that's where in the calcine AM is a green fluorescent cell. It says that the cell is alive. As one of those indicators we talked about earlier. And you can see that the background has decreased, and so now it's darker. And in the MitoTracker red, when they use the red channel background suppressor, they've made it so you can actually see those healthy mitochondria being labeled. And then in the blue channel, again, you can see that it cuts way down on the background. Hopefully you can still see that the nuclei are labeled on your computer screen. So that's something that you would use for live cell imaging to reduce your background. If you're having problems with background and you're doing fixed cell imaging, then a possible thing to do if you've exhausted everything else is to look into using signal enhancer. That's one of those ready probes reagents I talked about earlier that's in a dropper bottle. And you put it on your fixed and perm cells before the blocking, or you could actually use it after the blocking as well. But generally we recommend you put it on before you do your blocking step, and you'll see some help there where it'll reduce the background by quite a bit, and then you might be able to actually see those dimmer signals. So then while we're talking about fixed cells, another common problem that I mentioned earlier is that when you are doing your imaging experiment, you may have noticed that you can sometimes see what we refer to as photobleaching. And it's kind of related to phototoxicity, but instead of the light causing problems for the cells and causing them to die, the light actually causes the fluorophore to dim. And that's from, the molecule can only fluoresce so many times, and so then when the light is shown on it for too long or too many times, it starts to dim. And what you can do to get around that in your fixed and perm cells is to use a mounting media that has antifades in it, which prevent fading or photobleaching. And one that's pretty nice to use is ProLong Diamond. And the nice thing about it is that it also preserves the GFP signal in cells. And you can see that in that middle panel there, that the GFP signal doesn't go away through repeated scans, and it stays nice and bright because it's protected, along with other fluorophores as well. So that can be something to look at using if you're running into problems with photobleaching in your fixed and perm cells. If you have issues, excuse me, if you have issues during live cell imaging, it's a little trickier because a lot of those antifades that prevent photobleaching also can be very toxic to cells. But there is something that we offer called ProLong Live, as opposed to ProLong Diamond, which I just mentioned, and it's to help with photobleaching in a live cell experiment. And what you can see here is that the HeLa cells are expressing a mitochondria that's been labeled with GFP, and you can see that the ProLong Live in the media will help prevent that GFP from photobleaching compared to complete media shown underneath. And it basically increases the amount of time that you can image your GFP expressing cells, live cells, by about 50%. So I hope that this seminar has been helpful. If you want similar information and information about reagents and protocols, we do offer some free apps for your mobile devices, and I'm showing you the URL at the bottom there, or you can just search cell imaging in the application stores. And then another resource that's there for you is at www.thermofisher.com/imagingbasics. And that has a lot of information. Some of it I've covered here about different things if you're new to fluorescent imaging that you might want to know about. And so that's there to use. And with that, I'll go over the key takeaways. And so keys to successful imaging experiments, the practical considerations that the title alluded to: reduce your background. Do everything you can to make your signal high and your background low. And if you make that, the signal that which is what you want to see, and the background, which you don't want to see fluorescently labeled, as big as possible, you'll have the best outcome for your experiment. You also, if you're doing live cell imaging, you need to keep your cells really happy with environmental controls, because you can't just pop them on that icy bicycle and expect them to pedal away. And then lastly, you want to pick bright fluorophores that give you a nice bright signal so that you can use as little light as possible for excitation. And then you want to excite at the most optimal wavelength for the fluorophore you're using, which means match your filters to your fluorophore, make sure your instrument's functioning properly. And with that, I'll close, and I believe that you can ask questions in the Q&A. And thank you for attending.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them into the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question: which of these reagents would work in tissues?
That's always a very common question, and just like most of biology, the answer is it depends, which is never what people want to hear, but it really depends on what reagent you are looking to use. Some of them can work well in tissue, and it's really going to be tissue dependent. The one that always works in almost every tissue that people have tested it in was ClickIt EDU and ClickIt Plus. Those are the things that label proliferation, and those work very well in tissue. Other dyes that we mentioned here, like the trackers and things like that, it'll be really dependent on the tissue itself.
The next question: does CellEvent come in any other color besides green?
I think the question was, does CellEvent come in any color besides green? And the answer is, unfortunately, no, not right now. CellEvent is the probe that was for Caspase activity in the apoptotic pathway. And we know that our customers really want it in other colors, and the chemists and biologists here at Molecular Probes are working as hard as they can, and as soon as we have something that we think does a good job, we will be selling it. But right now the one that works the best is just available in the green color.
Great. I would like to once again thank Trillium Blackmer for her presentation. Do you have any final comments?
No, I don't have any final comments. Just be sure to check out those web resources, and you can always call customer support. That phone number was given in the beginning of the slide if you have questions about Molecular Probes products.
Today's webcast will be available for on-demand viewing until June 2017. You will receive an email from Labyrinth alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.